This is our second podcast in our series on bone. In this podcast, we're going to focus on the major cell types associated with bone. And so let me go back to this cartoon that we showed you earlier. We talk about the mesenchymal stem cell, which will give rise to the osteoprogenitor cells. The osteoprogenitor cells will differentiate into osteoblasts. The osteoblasts, when they're trapped in lacunae, will become osteocytes. We talk about periosteal cells and endosteal cells, sometimes called bone lining cells. These cells are similar to the osteocytes with the exception that they're not really trapped in lacunae. We also talk about the granulocyte monocyte precursor in bone marrow. These cells give rise to some of the white blood cells like neutrophils, eosinophils, basophils, monocytes, but they can also, under the right stimulations, these precursors give rise to inactive osteoclasts, which will differentiate into the active osteoclasts. So let's start with the osteoprogenitor cells. These cells are derived from mesenchymal stem cells, which are in the bone marrow. The mesenchymal stem cells are a renewable cell population. There's a transcription factor called core binding factor alpha 1, which will stimulate these mesenchymal stem cells to become the osteoprogenitor cells. These cells are found on the surfaces of bone. On the surfaces of bone, we talk about periosteal cells on the external surface, and on the internal surfaces of bone, we talk about endosteal cells. These are a flat or squamous type of cell. Be careful, they're not epithelial cells. We talk about them as a squamous type cell, but they're not epithelial cells. They're a mesenchymal type stem cell. These cells give rise to the osteoblast. The osteoblast is the differentiated bone-forming cell. This cell is synthetically very active. It secretes bone matrix, so primarily collagen 1 and other types of matrix proteins. It will also secrete an enzyme, alkaline phosphatase. Alkaline phosphatase is a useful clinical marker to look at osteoblast activity. The osteoblast, as it's secreting the matrix, is going to trap itself within lacunae. When the cells are trapped in lacunae, we consider them as osteocytes. The osteoblast and these osteocytes initiate calcification of the bone matrix. They secrete matrix-limited membrane vesicles. Calcium is accumulated in association with these vesicles. Secretion of alkaline phosphatase are involved in cleaving phosphate ions. This actually changes the local isoelectric point of the bone matrix. That causes the calcium phosphate crystals to form, and that leads to mineralization of the bone matrix. Osteoblasts themselves wind up being in communication with other osteoblasts through cell processes and gap junctions, and they also stay in contact with the osteocytes through gap junctions as the osteocytes are trapped in lacunae. The osteocyte is the mature bone cell that's enclosed by the bone matrix. The osteocytes maintain the bone matrix. They respond to mechanical stimuli. They can synthesize or degrade the bone matrix, so they play some role in calcium homeostasis. As we've suggested, they communicate with nearby cells via gap junctions. They may communicate with distant cells by various signaling molecules. Some of these signaling molecules can be nitrous oxide and perhaps glutamate. There are three functional states of osteocytes that we can describe. We talk about quiescent osteocytes. These are cells that are not active synthetically. We talk about formative osteocytes. These are synthetically active cells. They're almost like an osteoblast type cell, and we talk about resorptive osteocytes. These cells secrete matrix metalloproteinases, and through a process called osteocytic osteolysis, they can regulate calcium homeostasis. This osteocytic osteolysis is not playing a role in bone remodeling. That's primarily the role of the osteoclast, but resorptive osteocytes may help regulating calcium balance in the body. Here are electron micrographs of the osteocytes showing their three different functional states. On the left we can see a quiescent osteocyte. You see a little bit of rough ER. You see the bone matrix here. You see the densities around the osteocyte. Here are some little cell processes coming off. Here's a formative osteocyte. Lots more rough ER and even Golgi. And then the resorptive osteocyte. Again, some rough ER, perhaps some Golgi profiles, but you can begin to see profiles of lysosomes. What's very distinct on the resorptive osteocyte, look at the bone matrix density around both the quiescent and formative osteocytes. Look at the lack of density around the resorptive osteocytes, suggesting that this cell is actually maybe breaking down some of the bone matrix. Here are some bone lining cells at a histological level. These are derived from osteoblasts. These are cells that are covering the surface of bone when the bone is not remodeling itself. So these cells function like osteocytes, but they're not trapped in lacunae. They're on the cell surface. Here's an osteocyte here trapped in a lacunae. Very similar type of cell, but the bone lining cells are not trapped in lacunae. They can be either on the external or internal surfaces of bone. So here you can see them at the electron microscopic level with some cytoplasmic processes between them. And here at a higher magnification, you can see the gap junction between some of these bone lining cells. A very similar type view would be seen with the osteocytes connecting with each other within canoniculi between lacunae. Now, as we said, the osteoclast is responsible for bone resorption. Osteoclasts are derived from fusion of monocyte precursors in the bone marrow, so they're multinucleated cells. You can see the osteoclast here as a multinucleated cell tucked into this bone spicule. These cells secrete acid phosphatase. The differentiation of osteoclasts is a little bit complicated. Here is the common progenitor cell, the granulocyte monocyte progenitor cell, which is actually developing from a common myeloid stem cell in the bone marrow, and this progenitor cell is giving rise to the osteoclast precursor cell. Well, it turns out that osteoblasts secrete cytokines that will stimulate stromal cells to influence osteoclast precursors to respond to rank ligands. This rank signaling stimulates osteoclast differentiation. Here would be the inactive osteoclast, which will eventually differentiate into the active osteoclast. This cartoon shows how we think the osteoclasts function. Here's some histological views. The osteoclasts, as we said, are multinucleated cells on bone surfaces. So at this low power, you can see some osteoclasts like this. Here's a little bit higher power. You can see the osteoclasts. The osteoclasts resorb bone tissue by releasing protons and lysosomal hydrolases into a very constricted microenvironment in the extracellular space. This constricted microenvironment is given the name Hausipp's lacunae, and what you can think of is this osteoclast almost forming a tight seal around the edge of the bone. You can see these cytoskeletal elements helping to form the tight seal with integrins. You can see the protons that are being generated with carbonic anhydrase and secreted into the extracellular space. Chloride ion channels play a very important role in maintaining the electron neutrality of these membranes. These digestive proteins are released. The bone matrix is digested and it's reabsorbed into these osteoclasts. And at the same time that this is occurring, of course, breaking down the bone matrix, you can be regulating blood calcium. As you know, parathyroid hormone can increase osteoclast activity. That may cause blood calcium to rise. On the other hand, calcitonin will decrease osteoclast activity, and that can lower blood calcium.